In this module, we are going to talk about microbial control. We know that bacteria do not need much to grow and survive. A good carbon source, a nitrogen source, and water are mainly what they require. Hence, they grow almost everywhere in all those places where we would actually prefer them not to grow. They grow on and in our bodies and cause diseases, destroy crops by infecting them with diseases, bring about food spoilage, contaminate our pure cultures, and can be in fact a source of nuisance to us. So we are constantly in need of methods to keep these microbes at bay and to control them. Equipped with our knowledge of microbial growth, it is easier to control microbes. So we'll focus now on the different methods of microbial control. Controlling microbes can mean killing the microbes present or inhibiting their growth. Those substances which kill microbes are called bactericidal agents, and those which inhibit their growth are called bacteriostatic agents. When we are targeting at controlling microbes, we mainly are interested in cell death. But the cell death in a population does not occur instantly. Instead, just as cells grow exponentially, they also die exponentially. Whatever treatment the bacteria are subjected to, they die at an exponential rate. Exponential death means that they die at some fractional rate per unit time. If 50% of the microorganisms in a population die every minute, then after 2 minutes, there will be 25% still alive. After 3 minutes, there will be 12.5% still alive. And after 4 minutes, there will be 6.25% still alive, and so on. If the logarithm of the population number remaining is plotted against time of exposure of the microorganisms to the agent, a straight line will result, as can be seen in the figure on your screen. The death of a given microbial culture depends upon a few factors which have to be considered while selecting the control method. The first factor is the degree of contamination, which is also called the microbial load. The more the microbial population, the more time it will take for sterilization. A high population load will require more time as compared to a low population load as shown in this figure. The second factor is the susceptibility of the microbes to the antimicrobial agents. It is necessary to carefully select an antimicrobial agent because some microbes are resistant or less susceptible to certain antimicrobial agents. For example, it is no use trying to kill Deinococcus radiodurans using radiations because it is resistant to radiations. The third factor is the time of exposure. Time is a factor relative to other factors. More the microbial load, more is the time required for killing the entire bacterial population. Also, more the concentration or intensity of the antimicrobial agent, less is the time required. For example, when 1% phenol is used as antimicrobial agent, it takes 20 to 30 minutes to kill the bacterial population, but when 5% phenol is used, it takes just 2 to 3 minutes. The next factor is concentration or intensity. The concentration of the chemical agent and the intensity of the physical agent determines the time required to kill a microbial population. For example, we just saw that 5% phenol takes lesser time to kill the microbial population as compared to 1% phenol. But sometimes the case is just reverse. Like 70% ethanol is more effective than 100% absolute ethanol because the presence of water enhances the activity of alcohol. The next factor is physiological condition of microbial population. The effect of the antimicrobial agent depends upon the physiological state of the microbial population. Young, actively growing cells are killed more easily than old, dormant, non-dividing cells. The next factor is the type of the microorganism. The effect of the antimicrobial agent differs with the type of the microbes like bacteria, fungi, viruses, etc. The activity differs for gram-negative and gram-positive bacteria within a group too. Some species are more resistant to antimicrobial agents than other bacteria. Furthermore, bacterial as well as fungal spores are more resistant than the vegetative cells. The next factor is environmental factor. Many environmental conditions like temperature and pH influence the effect of antimicrobial agents. For example, the killing action of ethanol is greater at higher temperature than at lower temperature. Also, the presence of organic material like dirt, oil, saliva, blood, fat, etc. might interfere with the penetration of heat, chemicals and radiations and also inactivate the chemical disinfectants. Let us first discuss some commonly used terms in microbial control. The first term is sterilization. It is defined as the process where all the living microorganisms, including bacterial spores, are killed. There is no degree of sterilization. An object is either sterile or not. Sterilization can be achieved by physical, chemical, and physicochemical means. The second term is disinfection. Disinfection is the process of elimination of most pathogenic microorganisms on inanimate objects. Disinfection can be achieved by physical or chemical methods. Chemicals used in disinfection are called disinfectants. Different disinfectants have different target ranges. Not all disinfectants can kill all microorganisms. The next term is decontamination. It is the process of removal of contaminating pathogenic microorganisms from the articles by a process of sterilization or disinfection. It is the use of physical or chemical means to remove, inactivate or destroy living organisms on a surface so that microbes are no longer infectious. The next term is sanitization. Sanitization is the process of chemical or mechanical cleansing applicable in public health systems, usually by the food industry. The next term is asepsis. Asepsis is the employment of techniques to achieve microbe-free environment. Antisepsis is the use of chemicals on skin or mucous membranes to keep it free of pathogenic microorganisms. An antiseptic is a substance that prevents the growth and reproduction of various microorganisms on the external surfaces of the body. Bacteriostasis is a condition where multiplication of the bacteria is inhibited without actually killing them. Bactericidal is that chemical that can kill or inactivate bacteria. Such chemicals may be called variously depending on the spectrum of activity such as bactericidal, virucidal, fungicidal, microbicidal, etc. Let's now talk about the common methods of microbial control. We'll be talking about the physical agents, which would include heat, radiations, and filtration. We'll also be talking about chemical agents, which will include phenols, alcohols, halogens, and gaseous agents in the coming slides. Moving ahead, let us talk about the physical methods of control. The physical methods of microbial control include temperature, irradiation, filtration, dehydration, etc. Let us first talk about temperature. We already know the influence of temperature on the growth of microbes and how every organism has its own optimum growth temperature. Naturally, temperature above the optimum would be lethal and can be used to kill the microbes. So also, temperatures below optimum can be used to retard the growth of microbes. High temperature is the most widely used and reliable method of sterilization, including involving destruction of enzymes and other essential cell constituents. The efficiency with which high temperature is able to inactivate microorganisms is dependent upon the degree of heat, the exposure of time, and the presence of water. The action of heat will be due to the induction of lethal chemical events mediated through the action of water and oxygen. In the presence of water, much lower temperature time exposures are required to kill microbes than in the absence of water. Both dry heat and moist heat are used for sterilization. When trying to develop an effective heat treatment for a food or culture medium, it is often necessary to quantitatively determine the rate of death. Initially, thermal death point was used to express this. It was defined as the lowest temperature at which the bacterial population is killed in 10 minutes. This was then replaced by the concept of thermal death time. The thermal death time is the time it takes for a population of cells to reach effectively zero at a given temperature. But it is virtually impossible to reach zero value. Hence, decimal reduction time or D value is more accepted these days. The decimal reduction time is a measure of heat sensitivity of a microbe and is defined as the length of time it takes for the viable population to decrease tenfold or 1 log cycle at a given temperature. Hence, the time or the dose it takes to reduce 1000 microbial cells to 100 cells is the t-value. This can be determined by incubating a culture at a lethal temperature and periodically determining the number of surviving microbes in the sample by viable plate count. This is called the survival curve method. The log of the viable plate count is plotted against time and a straight line is drawn through these points. The negative reciprocal of the slope of the line is the decimal reduction time. The value is only valid for the microbe under study at that temperature and in the medium used. For example, the D value at 72 degrees of 1 minute means that for each minute of processing at 72 degrees, the bacterial population of the target microorganisms will be reduced 90%. In this figure, the D value is 14 minutes, 40 minus 26, and would be representative of a process at 72 degrees. Once D values have been determined for at least 3 temperatures, the Z value can be determined. The Z value measures the sensitivity of a microbe to a change in temperature and is defined as the amount of temperature increase necessary to decrease the D value tenfold. To determine the Z value for a microbe, at least 3 D values are plotted against its temperature and a line is drawn through the points. The Z value is the negative reciprocal of the slope of the line. With a Z value in hand, it is possible to predict the microbe's death rate in the specific medium at any lethal temperature. D values, TDT and Z values are very useful in the food industry where one wants the treatment to appropriately reduce the microbial population with minimum negative effect on the food itself. Let us now talk about the types of heat treatment. Dry heat. The first is flaming. This is one of the most ancient methods of killing microorganisms and is commonly used in laboratories nowadays. Here the article to be sterilized is passed over a Bunsen burner's flame but not heated to redness. Articles such as scalpels, inoculating loops, mouth of test tubes, flasks, glass slides, etc. are passed through a flame a few times. Even though most of the vegetative cells are killed, there is no guarantee that spores too would die. In this figure, you can see the flaming of an inoculating loop. The next method of dry heat is hot air oven. Dry heat is sometimes recommended for sterilization, where the moist heat can spoil the materials like oils, powders, etc. The apparatus used is a hot air oven and can be routinely used for sterilizing glasswares like petri plates, pipettes, etc. A 1 to 2 hour exposure at 160 to 180 degrees is enough to bring about sterilization. Since air is a poor conductor of heat, even distribution of heat throughout the chamber is achieved by a fan. The heat is transferred to the articles by radiation, conduction, and convection. The oven should be fitted with a thermostat control, temperature indicator, meshed shelves, and must have adequate insulation. Articles to be sterilized must be placed at sufficient distance so as to allow free circulation of air in between. Mouths of flasks, test tubes, and both ends of pipettes must be plugged with cotton wool. Dry heat is an effective method of sterilization of heat-stable articles. The articles remain dry after sterilization. This is the only method of sterilization of oils and powders. However, there are a few disadvantages. These are, since air is a poor conductor of heat, hot air has poor penetration. Cotton wool and paper may get slightly charred. Glassware may become smoky, and it takes longer time compared to autoclave. Let us now move on to moist heat. Boiling is the most common method. Boiling water kills most vegetative bacteria and viruses immediately, but some bacterial spores are resistant to boiling and survive. Hence, this is not a substitute for sterilization. Certain bacterial toxins such as staphylococcal enterotoxin are also heat resistant. The killing activity can be enhanced by addition of 2% sodium bicarbonate. When absolute sterility is not required, certain metal articles and glasswares can be disinfected by placing them in boiling water for 10 to 20 minutes. Before the advent of the disposable plastic syringes in hospitals and dispensaries, the glass syringes and needles were disinfected by boiling. Boiling is also used as a household method of disinfecting milk bottles of infants. The second method of using moist heat is Arnold steam sterilizer. The Arnold steam sterilizer was widely used in US and is based on the principle of live steam. As shown in the figure, it consists of a cylinder of tin or copper with a cover, which is enclosed in a movable cylindrical outer cover or hood. The inner cylinder has a perforated bottom, as you can see, through which the steam may enter. The steam comes from the small chamber below the copper bottom, which contains water which is boiled. The peculiarity of this type of sterilizer is that the steam which escapes from the sterilizing chamber condenses beneath the outer cover and falls back upon the chamber in which the steam is generated. Thus, this sterilizer uses live steam, which has a temperature of 100 degrees, and a single exposure for 90 minutes can satisfactorily sterilize the materials. The next method is sterilization. Certain spore-forming bacteria can resist the action of steam for hours. Such prolonged exposure to steam might be harmful to the culture media containing gelatin, milk and sugars. Hence, fractional or discontinuous sterilization or tyndallization is used. Here the medium is exposed to steam for 3 consecutive days. The idea behind it, as proposed by John Tyndall, was that the culture medium to be sterilized might contain bacteria in their vegetative stage as well as in the spore stage. So the media to be sterilized is boiled for a period of time, say 30 minutes, and then cooled for 24 hours and then boiled again. This boiling is repeated 3 times. What is achieved during the period of intermediate cooling? Boiling is enough to kill vegetative cells, but not the spores. But during boiling, the spores are activated, and during the cooling period, they germinate to form vegetative cells, which are in turn destroyed by the next boiling phase. The most common method in every microbiology lab for using moist heat, however, is autoclave. It is the most practical method of sterilization. The autoclave was invented by Charles Chamberlain in 1884. It is a closed device which works on the principle of saturated steam under pressure, the same as that of the pressure cooker. It uses moist heat to kill not only all the vegetative cells but also the endospores. Moist heat is more effective than dry heat because it has more penetrating power. It is supposed to act by degrading nucleic acids and denaturing proteins. At a pressure of 15 pounds inside the autoclave, the temperature is said to be at 121 degrees. Exposure of articles to this temperature for 15 minutes is enough to bring about sterilization. Let us now talk about the construction and working of an autoclave. A simple autoclave has a vertical or horizontal cylindrical body with heating elements, a perforated tray to keep the articles, a lid that can be fastened by screw clamps, a pressure gauge, a safety valve, and a discharge tap. The articles to be sterilized must not be tightly packed. The screw caps and cotton plugs must be loosely fitted. The lid is closed but the discharge tap is kept open and the water is heated. As the water starts boiling, the stream drives air out of the discharge tap. When all the air is displaced and the steam starts appearing through the discharge tap, the tap is then closed. The pressure inside is allowed to rise up to 15 pounds per square inch. At this pressure, the articles are held for 20 minutes, after which the heating is stopped and the autoclave is allowed to cool. Once the pressure gauge shows the pressure equal to that of the atmospheric pressure, the discharge tap is opened to let the air in. The lid is then opened and articles are then removed. There are certain precautions which have to be taken while using an autoclave. Firstly, the autoclave should not be tightly overpacked because steam needs to circulate freely in the chamber and contact each and every object. Secondly, if the chamber is not totally free of air, then it will not reach a temperature of 121 degrees even if it shows a pressure of 15 pounds. And remember, it is not the pressure which kills the microbes but the high temperature which is achieved by steam under pressure. Thirdly, when the pressure is released, it should be done slowly or else the contents inside will be thrown upwards. Also, since moist heat is used, heat labile objects cannot be sterilized using an autoclave. There are certain chemical, biological and physical indicators that can be used to ensure that an autoclave has operated for the prescribed time at the correct temperature. Chemical indicators consist of a sterile autoclave tape which changes color once the correct conditions have been met. This indicates that the object inside the package or under the tape has been autoclaved sufficiently. Biological indicators include a culture tube containing sterile medium and spores of heat-resistant bacterium Bacillus stereothermophilus. If the autoclave did not reach the right temperature, the spores will germinate and their metabolism will change the color of a pH sensitive chemical. Physical indicators often consist of an alloy designed to melt only after being subjected to 121 degrees for 15 to 20 minutes. If the metal has melted, the change will be visible. Moving ahead to pasteurization. This is a process whereby milk and food are held at temperatures below their boiling points for a specified time for killing harmful organisms such as bacteria, viruses, protozoa, molds, and yeast. The process was named after its inventor, French scientist Louis Pasteur. Milk pasteurization. Franz-born Soxhlet in 1886 first adapted pasteurization for preserving milk. Two methods are very commonly used to pasteurize milk. High temperature short time method. It's also called flash pasteurization and is by far the most common method. It involves holding the milk at temperature of 72 degrees for at least 15 seconds. The high temperature short time pasteurization standard was designed to achieve a 5 log reduction in the number of the viable microorganisms in milk. This is considered adequate for destroying almost all yeast, mold, and common spoilage bacteria, and also to ensure adequate destruction of common pathogenic heat-resistant organisms, particularly Microbacterium tuberculosis, which causes tuberculosis, and Coxiella burnetii, which causes Q fever. The next method is ultra high temperature, which involves holding milk at a temperature of 138 degrees for 2 seconds. Ultra high temperature treated milk does not require refrigeration and can be safely stored at room temperature. The next physical method of microbial control is irradiation. Radiations are mainly of two types. Ionizing radiations. These have enough energy that they can act on the target molecules, remove electrons and form ions. X-rays, gamma rays, and electron beams are examples of ionizing radiations. Their wavelengths are smaller than 1 nanometer. The second type of radiations are non-ionizing radiations. These cannot form ions. UV rays are a type of non-ionizing radiations. Their wavelength is longer than 1 nanometer and they are less effective as compared to ionizing radiations. Ionizing radiations like X-rays and gamma rays collide with various molecules in the microbes and produce hydroxyl and hydride radicals. These highly reactive molecules are capable of degrading DNA and proteins and hence the subsequent death of the microbial cells. UV radiations bring about damage in the DNA. They cause the formation of covalent bonds, particularly two adjacent thymines will undergo dimerization to form what is called as thymine dimer. The two thymines undergoes bond formation between the fifth and the sixth positions to form what is called a cyclobutyl ring. The formation of the dimers causes a distortion in the DNA strand and prevents its replication and transcription and brings about the death of the cell unless the damage is repaired. The amount of radiation necessary to kill a microbe is species specific and depends on a variety of factors. Gram-negative bacteria like Pseudomonas, Escherichia, Shigella and Salmonella are more sensitive to radiation than most gram-positive bacteria. Spores of Bacillus and Clostridium are even more resistant due to special proteins in the spores that are thought to protect the DNA. The use of radiations for sterilization is measured in grays. 1 gray is defined as absorption of 1 joule of radiation per kilogram. In general, microorganisms are more resistant to ionizing radiations than higher organisms. For example, the amount of energy required to reduce the bacterial load by 10-fold is at least 200 grays, whereas the lethal dose for humans is just 10 grays. Radiation is currently used as a sterilization practice in food and drug industries. Food and Drug Administration has approved the use of radiation for sterilization of surgical supplies, disposable labware, drugs, and even tissue grafts. The third physical method of microbial control is filtration. Though heat is the most common and easy method of sterilization, not all materials can be sterilized by it. Heat-sensitive liquids like hormones, vitamins, etc. and gases are sterilized by filter sterilization. Filtration physically removes the microbes by employing a filter or a membrane which has pores too small to allow the passage of microbes but large enough to allow the passage of liquid or gas. We know of microbes which have a diameter as small as 0.3 micrometers. Hence, the pore size of the membranes commonly used for microbiological purposes is 0.2 micrometers. But a filter of this pore size would not remove viruses because they are smaller in size. A filter system with sufficiently small pores to remove viruses would have an extremely slow flow rate due to the viscosity of water. Because viruses are not removed, filtration cannot be technically considered a form of sterilization, although it is common to refer to the process as filter sterilization. Let's talk about the types of filters. There are 3 major types of filters. The first are what are called as depth filters, which are the oldest type and are made of overlapping layers of fibrous sheets of papers, asbestos or glass fibers. This type of filters are mainly used as pre-filters and they efficiently remove the larger particles so that they do not clog up the more sophisticated filters during the final filter sterilization process. The most common type of filtration method used in modern microbiology labs are membrane filters. These are made from high tensile strength polymers of cellulose acetate, cellulose nitrate, polycarbonate, polyester, polypropylene or polysulfone. These polymers appear as a thick mesh of interconnected strands with rather precisely defined spaces between the polymers. The third type of filters are nucleopore filters. These filters are created by exposing a very thin polycarbonate film to nuclear radiation, which creates areas of local damages in the polymers. The filters are held in special holders or holding apparatus of glass or metal. The solution to be filtered is forced through the filter with the help of a vacuum or pressure using a syringe or a peristaltic valve. The filtered solution is collected in a previously sterilized container. The filtering apparatus and filters are generally sterilized separately and assembled aseptically, or they are available pre-sterile. We will be talking about the chemical methods of control. Quite often, chemical agents are very widely used for microbial control. This is because their use is convenient as well as practical. For example, wiping the tabletop with a disinfectant before a microbiology experiment or wiping the skin with an antiseptic before an injection. A careful selection of the chemical agent depends upon a number of factors. The first is the type of the material to be treated. It is always better to have a complete knowledge of what is to be sterilized. One cannot use an antiseptic for sterilizing a tabletop or a disinfectant for sterilizing skin. This is because the disinfectant, being stronger, can cause harm to the tissues of the skin, and the mild antiseptic, though suitable for the delicate tissues of the skin, might be completely useless on an inanimate surface. The second factor is the type of the microbes to be removed. It is again better to have some idea about the type of the microorganisms which might be predominant on the material to be sterilized. This is because not all chemical agents are equally effective against bacteria, viruses, fungi, and other microorganisms. Among the bacteria, a particular chemical agent might be more effective against gram-positive bacteria than gram-negative. Hence, a careful selection of the chemical agent is a must for effective disinfection. The third factor is the influence of environmental conditions. We know that environmental parameters do have an effect on the growth of microbes and hence would also play an important role in controlling the growth of the microbes. While selecting a chemical agent, one should keep in mind the effect of these parameters on the agent so that it can be used under the most favorable conditions. An ideal chemical agent should have some of the characteristics stated below to be an effective and safe chemical agent. The first is antimicrobial activity. Ideally, a chemical should be effective against a wide variety of microbes and that too at a very low concentration. Toxicity to humans. At the same time, it should be totally harmless and non-injurious to the humans and other living things. In practice, though it is very hard to achieve a balance between low toxicity and effectiveness. The third factor is solubility. The chemical agent should be readily soluble in water and other solvents so that it is easily applicable. The next factor is stability. The chemical agent should be stable upon storage and should show no decrease in its activity upon standing. The next factor is the ability to penetrate. The chemical agent should have an ability to penetrate through the surface so that it has a deep reaching action, or else its action would only be at the surface level. The next factor is odor. It should be either odorless or it should have a pleasant odor. The next factor is non-corroding. The chemical agent should not have any corrosive effect on the metals, nor should it have any harmful effect on the fabrics, etc. The last factor is reaction with organic matter. If there is considerable amount of organic matter along with microbes, some chemical agents have a tendency to react with the organic matter, and in such cases there will be very little of it left to react with microbes. Hence, an agent which does not react or reacts less with organic matter is preferred. The important chemical antimicrobial agents used as disinfectants or antiseptics are phenols, alcohols, halogens, surfactants, heavy metals, and gaseous agents. Let us first talk about phenols and phenolic agents. Phenol has a history of being used by Sir Joseph Lister way back in 1867 to reduce the risk of surgical infections. Phenol also has the credit of being used as a standard against which the antimicrobial activity of other disinfectants is compared by using a technique called phenol coefficient technique. Phenol refers to the class of aromatic compounds having a hydroxyl group as well as other substituent groups on a 6 carbon benzene ring, as can be seen on your screen. Phenol is bacteriostatic at concentrations of 0.1 to 1% and is bactericidal or fungicidal at 1 to 2%. A 5% solution of phenol kills anthrax spores in 48 hours. The bactericidal activity is enhanced by EDTA and warm temperatures and decreased by alkaline medium, lipids, soaps and cold temperatures. Concentrations greater than 0.5% exert a local anesthetic effect, whereas a 5% solution is strongly irritating and corrosive to tissues. Oral ingestion or extensive application to skin can cause systemic toxicity manifested primarily by the central nervous system and cardiovascular effects, which may lead to death. Phenolics are the phenol derivatives which have been chemically modified by the addition of some organic functional groups. Phenols and phenolic agents like crisols, xylenols, orthophenylphenols are very effective disinfectants. A 5% to 6% aqueous solution of phenol is effective in killing vegetative cells but not spores. The commercially available Lysol is a mixture of phenolics. Bisphenols, a combination of two phenol or phenolic molecules, are widely used in modern disinfection and antisepsis. For example, orthophenylphenol is used in Lysol, hexylresorcinol is used in mouthwash and throat lozenges. Triclosan used in a number of consumer products. Hexachlorophene has a strong bacteriostatic action against many gram-positive organisms and was used as a popular household bisphenol, but it was found to cause brain damage in infants and now it is used only in response to severe staphylococcal infections. The mode of action of phenols and phenolics mainly kill the microbes by denaturing the proteins, inactivating enzymes, and disrupting the cell membrane. They are advantageous because they remain active on the surface long after application as compared to volatile halogens or alcohols. Their drawback is that they have a very disagreeable odor and also cause skin irritation. Let us move on to what is called as the phenol coefficient determination. This technique is used to determine the effectiveness of the test disinfectant as compared to that of the phenol under similar conditions. The procedure for this includes that a series of dilution of phenol and the experimental disinfectant are prepared in sterile tubes. The test organism, which may be E. coli or S. aureus, is inoculated in each of the dilution tubes and then they are incubated for 5, 10 and 15 minutes. 100 microliter is then transferred from each tube to a fresh sterile broth tube and then it is incubated at 37 degrees for 48 hours. The highest dilution of the test antimicrobial agent and phenol that effectively kills the test organism within 10 minutes but not within 5 minutes exposure are used to compare the phenol coefficient. The turbidity indicates growth and is labeled as positive, and no turbidity indicates that there is no growth and it is labeled as negative. In the table, you can see the values for both the disinfectant as well as phenol. Here, the phenol dilution of 1:70 failed to kill the test organism in 5 minutes, but killed it in 10 minutes. The test infectant gave the same results at the dilution 1:200. Therefore, the phenol coefficient is calculated as the highest dilution of the test infectant upon the highest dilution of phenol. This gives us the result as 200 upon 70, which equals to 2.8571. Thus, the disinfectant is more effective than phenol. Agents with a phenol coefficient greater than 1 are more effective than phenol. The next chemical agent that we are going to talk about is alcohol. Alcohols are quite effective against bacteria, fungi, and some viruses, but not against spores. Their potency increases, but water solubility decreases with the chain lengths. Ethyl alcohol is the most commonly used alcohol. Methyl alcohol is less effective as compared to that of ethanol. Furthermore, it is poisonous and hence not used as a disinfectant. The higher alcohols like propyl, butyl and amyl are also germicidal than ethyl alcohol. Alcohol is routinely used to disinfect the skin before injections or drawing blood. It is also used to disinfect thermometers and small instruments like forceps, scalpels, etc. by immersing them in alcohol for 10 to 15 minutes. Surprisingly, 70% ethanol is more effective than absolute alcohol. This is because absolute alcohol has a strong dehydrating agent effect which causes the pores of the cell to close and prevents the entry of the alcohol inside the cell. This causes only the surface proteins to get coagulated, which is just bacteriostatic. When the conditions for the cell become normal, the cell will start growing again. On the other hand, 70% alcohol allows the alcohol molecules to penetrate inside the cell and cause complete coagulation of proteins inside the cell, which ensures cell death. The mode of action of the alcohols is that alcohols act as an antimicrobial agent by denaturing the proteins. They are solvents for lipids and hence also dissolve the membrane lipids and cause damage to the cell membrane. Alcohols are dehydrating agents and can bring about dehydration of the cells. The use of alcohol as a surface disinfectant is probably more due to the mechanical removal of microbes than the chemical action on them. The next chemical agent is halogens. A halogen is any of the 5 elements chlorine, fluorine, bromine, iodine and astatine belonging to the group 7A of the periodic table. The halogens, iodine and chlorine are important antimicrobial agents. They can be used alone or combined with some other elements. Let us first talk about iodine. Iodine is one of the oldest antimicrobial agent and has been used for more than a century now. It is used as a skin antiseptic and is effective against bacteria, fungi and some viruses. Iodine is not very soluble in water but is soluble in alcohol and aqueous solutions of sodium or potassium iodide. It finds its use as tincture of iodine, which is 2% or more iodine in water ethanol solution of sodium or potassium iodide. Although it is an effective antiseptic, it has its drawbacks of causing damage to skin, leaving stains on the skin and causing skin allergies. More recently, iodine is used as an iodophore in which it is complexed with an organic compound. It releases iodine slowly and has the advantage of being long-lasting, non-irritating and non-staining. Betadine is a well-known example of an iodophore which is applied to wounds. It can destroy bacteria, fungi and some protozoa but not endospores. Iodophore solutions retain good antimicrobial activity at pH less than 4 even in the presence of organic matter. The mode of action is that it acts as an oxidizing agent and it might oxidize important protein and enzymes and account for cell damage. It is also suggested that it may act by halogenating tyrosine residues of the enzymes requiring tyrosine for their activity. The next halogen is chlorine. Chlorine is one of the most widely used disinfectants. It is used both in its gaseous form and also in its combined form. Chlorine gas has been used since ages for purification of water supplies. Chlorine gas is difficult to handle, hence its usefulness is limited. A number of chlorine compounds are available and they are easier to handle than free chlorine. These compounds are sodium and calcium hypochlorites. These compounds are used in dairy industry and restaurants to disinfect utensils. Hypochlorite has a history of being used by Sir Ignaz Semmelweis in 1846 to reduce the incidence of childbed fever. The practice was to wash hands and soak them in hypochlorite solution before examining patients. This practice had reduced the infections to 1/3. Another important group of chlorine compounds are the chloramines, which are chemical compounds of chlorine and ammonia. Examples are monochloramine, chloramine T, azochloramide, etc. They are used in wound dressings as skin antiseptics and also for treating water. They are less effective than hypochlorites, but they are more stable, release chlorine slowly, and are long-lasting and hence more preferred. Chlorine dioxide is a gas that can be used to disinfect large spaces. For example, it was used to treat the office buildings contaminated with anthrax spores after the bioterrorism attack of 2001 in US. The mode of action. The action of chlorine and chlorine compounds is mainly due to the formation of hypochlorous acid as can be seen in this equation. The hypochlorous acid formed is further decomposed to form nascent oxygen. The nascent oxygen is a strong oxidizing agent and acts on cellular components to bring about the destruction of microbes. Organic matter interferes with chlorine action by reacting with chlorine. Hence, excess of chlorine is added to ensure complete microbial destruction. The next halogen is bromine. Bromine is yet another halogen which is used for chemical control, but its use is particularly restricted to recreational and industrial water disinfection, including swimming pools, chlorine towers, etc. 2 to 4 mg per liter is enough over a wide temperature of 5 to 45 degrees and a pH of 6 to 9 range. The compounds of bromine are well-established broad-spectrum antimicrobials. In comparison to chlorine, bromine is considered less volatile and less toxic to aquatic life but it is very irritating. They are also less corrosive, have a lower toxicity profile, relatively safe to use, and easy to handle. The formation of bromate and haloacetic acids as byproducts of water disinfection are of concern because these compounds are known to be carcinogenic to humans. The presence of UV light and reducing agents can degrade bromine and decrease its overall efficacy. The use of methyl bromide is banned in many countries because it is a neurotoxin and it is also able to damage the ozone layer. The mode of action: when bromine is dissolved in water, hypobromous acid and hypobromite ion are formed, which are responsible for the antimicrobial activity. Bromine also reacts with ammonia or nitrogen compounds and produces bromamines, which too have an antimicrobial activity. Similar to other halogens, bromine oxidizes organic molecules like proteins, nucleic acids, and lipids, which results in cell death. Also, methyl bromide reacts with sulfhydryl groups of protein and enzymes, which leads to inhibition of cellular biochemical processes. The next halogen is fluorine. Fluorine too is a halogen and is one of the most reactive chemicals. Fluoride is claimed to reduce the incidence of dental cavities by reaction with tooth enamel. A number of different fluoride compounds like silicofluorides, stannous fluoride, and amine fluorides are used for water treatment, toothpastes, mouthwashes, etc. Sodium and potassium fluorides have been used as preservatives, but they are of limited use now due to their toxic effects. Let us now talk about heavy metals as antimicrobial agents. Many salts of heavy metals like silver, mercury, and copper are used as antimicrobial agents. The action of heavy metals on microorganisms is termed as the oligodynamic action. These metals are at very low concentrations exhibit antimicrobial activity. A small piece of heavy metal is placed in the center of the sterile nutrient agar plate, seeded with a test organism like Asorius, and the plate is incubated. Small quantities of metal diffuses into the agar and inhibits the growth of the test organism, as can be seen on your screen. The inhibition of the growth of the organism is seen as a zone of inhibition. Let us talk about these heavy metals in slight detail. Silver. In the olden days, a few drops of 1% silver nitrate solution was placed in the eyes of the newborn babies to protect from infection by Neisseria gonorrhoeae. A 0.5% solution is sometimes applied as a dressing on burns to reduce infection. Colloidal silver compounds which release silver ions slowly are bacteriostatic and have a more sustained effect. Today, silver nitrate has been replaced by antibiotic ointments which are less irritating and effective against pathogens also. Copper. Copper is not only bactericidal at low concentrations, but it also has an algicide and fungicide. It is used as copper sulfate in swimming pools and water supplies. Copper compounds are also used as preservatives in paints, cement, fabric, etc. to provide an antimicrobial barrier to surfaces where they are applied. Mercury is used as an antimicrobial agent since many years. It is used as mercury chloride in products like mercurochrome, merthiolate, and metafe. Mercury is combined with organic carrier compounds that reduce its toxicity. The mode of action: The antimicrobial action of heavy metals is due to their ability to react with the sulfhydryl groups of cysteine residues in the cellular enzymes. Further reactions can also lead to formation of hydroxyl radicals. This changes the configuration of the enzyme proteins, which in turn lose their biological activity. At high concentrations, they may also precipitate cell proteins, resulting in cell death. Positively charged ions have an affinity for negatively charged bacterial cell surfaces. Cell surface attack can lead to altered permeability and disruption of cell wall and membrane functions. Copper and silver ions also bind to phosphate backbone of DNA, leading to damage. Let us now talk about the gaseous agents as antimicrobial agents. There are many equipments and apparatus like plastic syringes, rubber gloves, plastic pipettes, plastic petri plates, which cannot be sterilized either by dry heat or moist heat because they are damaged by it. These are sterilized by gaseous agents. Large spaces like laboratories, operation theaters, and storage rooms are also sterilized using gases. When gaseous agents are used for sterilization, it is called fumigation. Chlorine and sulfur dioxide were used for fumigation earlier, but they are too poisonous for general purpose and hence they are no longer used. Presently, formaldehyde, ethylene oxide, and beta-propyolactone are among commonly used gaseous agents. Let us first talk about formaldehyde. This is a very irritating gas but can be generated by heating concentrated solutions of formaldehyde. Unless used at raised temperatures, it has a tendency to polymerize to form thin films of paraformaldehyde and hence its use is limited. But its killing effect, it's a irritating gas that can be generated by heating concentrated solutions of formaldehyde. Formaldehyde has a microbicidal effect, killing both the vegetative cells and spores. But its killing effect is greatly influenced by factors like humidity and temperatures. For best results, the temperature should be around 22 degrees and humidity at 70%. However, there are some disadvantages associated with the use of formaldehyde as a fumigant. These are: it has a low penetrating power and hence acts only at the surface. Secondly, it is very irritating to tissues and eyes. Thirdly, it is not very stable. And finally, the residual gas remains for a long time even after the aeration. The mode of action: it is a strong reducing agent. It combines with nitrogen containing macromolecules like proteins and nucleic acids and denatures them. The next gaseous agent is ethylene oxide. The compound ethylene oxide is a gas at room temperature. Its boiling point is 10.8 degrees Celsius, below which it is a liquid. In its pure form, it is very toxic when inhaled, irritating to the skin and eyes and highly inflammable. Hence, for commercial use, it is mixed with carbon dioxide or freon. For example, 10% ethylene oxide and 90% carbon dioxide is commercially available as carboxyl, or 20% ethylene oxide and 80% carbon dioxide is available commercially as oxyfumes. These preparations are not inflammable and the microbicidal activity is also not compromised. The use of ethylene oxide as a sterilizing agent is widespread because not only is it microbicidal but also sporicidal. It is routinely used for sterilizing heat and moisture sensitive materials in hospitals and laboratories. It is a better sterilant than formaldehyde because it is inexpensive and readily available. It has a deeper penetration power and it is quickly removed by airing and hence the residual gas does not remain for long. Commercially available apparatus for application of ethylene oxide is an autoclave which allows the chamber to be filled with the gas and which has controls for temperature and humidity. For practical purposes, 500 mg of gas per liter is used at about 58 degrees and a relative humidity of 40% for approximately 4 hours. Ethylene oxide chambers are called gas autoclaves, and they have become the chemical counterparts of heat and pressure based autoclaves for sterilization purposes. The mode of action: ethylene oxide acts by alkylation and introduction of an organic group in place of a hydrogen atom in various molecules like enzymes and proteins. For example, the hydrogen atom in a free carboxyl amino or sulfhydryl group is replaced by an alkyl group. Beta propyolactone. At room temperature, it is a colorless liquid which has a sweet but irritating odor. Its formula can be seen on your screen. It has a boiling point of 155 to 160 degrees. It is not inflammable like ethylene oxide, but at the same time, it causes irritation to eyes. It kills both the vegetative cells as well as spores, and hence it is a good sterilizing agent. A relative humidity of 70 to 80% and temperature of 25 degrees is required for maximum activity. It is more active than formaldehyde and ethylene oxide, but at the same time it has less penetration power. In aqueous solutions, it undergoes hydrolysis to form inactive beta-hydroxypropionic acid. Also, its prolonged use is said to be having carcinogenic effects, and all this has limited its use for practical purposes. The mode of action: its mode of action is similar to that of ethylene oxide. It also acts by alkylation action on enzymes and proteins. With this, we come to an end of our discussion on chemical agents which are used as antimicrobial agents. Hope you liked the content of this video lecture. Thank you.